Good afternoon. So first, thank to Urs and Alain for organizing this symposium and for you two for attending. So, okay, so yeah, by now you have seen this slide a few times. So I'm going to talk about EEG and, sorry about that, about EEG and FNIRS. So one provides a neuronal signal and the other, the FNIRS, will provide a hemodynamic signal similar to fMRI. So as you can see here, we'll use the EEG for the good temporal resolution it has, and the FNIRS for the relatively good spatial resolution. So you might want to ask, why doing FNIRS EEG? Because technically or methodologically, it can be a little heavy to do. So first, it requires, of course, the access to both equipment and also to high qualified personnel trained in both techniques, who are sometimes really hard to find. You also, well, it also takes longer installation time, which can be a big issue with children and babies. You also have to compromise on the head coverage and number of electrodes and optodes you will have. So, for example, if you want to have an international 1020 system with EEG, you might not end up with an exactly 1020 system. And for the NIRS-EEG, you might not be able to use as many optodes as you want because there are some electrodes into the way between the optodes. Also, there are no many combined FNIRS-EEG device helmet software on the market, so you may have to develop your own tools if you have more specific questions. However, using both techniques together, of course, will provide a very rich information from both the neuronal and hemodynamic signals. Also, adding the EEG to FNIRS will help to better identify movement artifacts, will help to characterize the participant's states of consciousness, which is sometimes really hard to identify in babies or in young children, and will help to identify epileptogenic activity, which is the topic of the talk. So overall, adding EEG to FNIRS will improve data quality. Another question you could ask would be, why not doing fMRI EEG? I focus mostly on pediatric population, so there are many advantages of FNIRS over fMRI in this population, and there are some other advantages, as all techniques has limitation and advantages. So the FNIRS is less costly, so it's relatively cheap compared to fMRI, so it opens doors to do studies in low or middle income countries. Also, the portable can be, the NIRS-EEG device can be portable, so there are now several companies developing really small device. As you can see here, this is the NIRS system, which is very portable. However, using the marketed system that are portable, you will have to compromise of the number of electrodes and optodes because they have very few, very small coverage. So because of that, several labs have developed their own device to answer their questions. So in collaboration with Mohamed Sawan and Frédéric Lesage at Polytechnique Montreal, they developed a FNIRS EEG device using 128 NIRS channels and 32 EEG electrodes. So it's quite small, so it's right here. And Ali Kassab, a PhD student with Frédéric Lesage and Daniel Nguyen, showed that these devices provide a very strong and stable EEG and NIRS signal while the participants were cycling. So we now use it in ICU in epileptic patients in Montreal. Another very good advantage of FNIRS is its tolerance to movement. So it's easy to use in children or in people with cognitive or behavioral limitations. So we can record if the children is moving because the fibers and electrodes will follow the head, and we can record during the participant is lying in a bed or is sitting on his parents' laps, or is, like you can see here, lying down is in parents' arm. So we can get pretty good signal even if the participant is moving, or for example, for babies having a pacifier, or if the participant is doing a language task out loud. So we can, even with the articulatory movement, we can get a pretty good signal. Also, it has a better temporal resolution than fMRI, so we can follow in time the brain activation. So this is an example here. I don't know if the movie will, no. There is a movie there, I don't know if it will work, but basically this is a participant who was doing a language task, a verbal fluency task, who was saying as many words as possible out loud belonging to a specific category. And obviously the movie is not working, but we can follow over the brain the activation. So first there is a Broca area activation, and then we see the recruitment of right homologous region and more posterior language area. So we can follow activation into the brain over time. We can also follow, for example, on a clinical point of view, epileptic seizure over time into the brain. And the main limitation of FNIRS compared to fMRI is its low spatial resolution compared to, it's pretty good, but it's not as good at fMRI. So it's about 1 centimeter over the cortex, and we are also limited by the shallow penetration of the light into the cortex. So it's not possible to record deep structure or subcortical structures. So it depends what is your research question, but it might be a very big limitation. So now I'm going to talk about the use of NIRS-EEG in epilepsy, mostly in children. A little background about epilepsy. So it's a brain disorder characterized by hindering the predisposition to generate epileptic seizures. That's the most frequent neurological disorder in children. The first choice treatment is pharmacotherapy. However, about 1/3 of patients are pharmacoresistant. So in this patient, we may consider surgery. And of course, before doing a brain surgery, we need to do all pre-surgical assessments to localize epileptogenic zone and the language function areas. So the pre-surgical assessment include many tests, for example, video EEG, structural MRI, neuropsych assessment, etc. And for several years now, epilepsy clinics around the world included fMRI and EEG, MEG to localize epileptic focus and language functions. However, although it works really well in adults, it's really hard to use in children, in young children, and in patients with moderate to severe cognitive and behavioral problem. So FNIRS has been developed as an alternative for children and patients with strong limitation. So this is an example. This is the first patient we did FNIRS-EEG to localize epileptogenic function. It was a 10-year-old boy with right frontal lobe epilepsy. And we did EEG and NIRS. We can see both signal here. And on EEG here, we can see there is an electrical seizure. And at the same time, in FNIRS, so here on X we have time in seconds, here we have changes of concentration in hemoglobin. Each line is one channel over the right frontal area, and the red line, the horizontal red line, is the duration of the seizure. So we can see that associated with the seizure, there is a huge increase of oxyhemoglobin and a small decrease of deoxyhemoglobin. So this signal allowed us to localize the epileptogenic zone in this patient. And this patient didn't have any intellectual disability. He had mild cognitive impairment, so we were able to do many other techniques with him. So we had more traditional techniques, PET, and also fMRI EEG, Pedro talked about it, and EEG, MEG. Both were done at McGill by John Gottman and Christophe Grova. And we can see that for all techniques, we have a good concordance between all the modalities. So this child got surgery almost 10 years ago and still seizure-free. We also use FNIRS-EEG to localize language function. So in a series of studies, we recorded children and adults with epilepsy who all did a language task, an expressive language task, and a receptive language task during a FNIRS-EEG recording. It is crucial to do EEG during FNIRS for language localization, because although FNIRS will allow to localize the language function areas, the EEG will allow us to have really clean FNIRS data. Because as soon as we're done with the recording, we want a neurologist to mark the EEG, to mark every epileptogenic activity on EEG, so spikes, seizures, to make sure that we remove these time sequence from the FNIRS signal, so we have a clean FNIRS signal. Because if we keep them, we might map the epileptogenic activity instead of language activities and mislead our interpretation. So this is another example of how what we see on EEG and FNIRS from Dang and Nguyen. So in this series of study, we compared the FNIRS results to other techniques like fMRI, VADA test, and intracranial recording. In all patients, we had access to these data, and we found a very good concordance between all the techniques. Also, we found conclusive FNIRS results in patients who could not undergo other procedures. Either they were too young or they were too anxious to do a VADA test or fMRI. So this is an example of what we see for the language activation using FNIRS. So it was a 9-year-old epileptic patient with left frontotemporal lobe epilepsy. This patient had a mental retardation, autism spectrum disorder, and anxiety issue. So because of his ASD, he had the stereotypical behavior, so he was moving like this during the recording. But we were able to have a pretty good signal, even if it was moving. So we can see during the expressive language task that there is a strong Broca area activation associated with the verbal fluency task. But at the same time, we have a significant activation also in the posterior language area, which usually typically will occur a little later, but in this patient it was simultaneous. So it shows probably a brain reorganization due to brain plasticity in this patient. There was also recruitment of right area, but it was significantly weaker than the left side. So it was left the dominance in this patient. So for several years now, we are, the FNIRS-EEG is part of the pre-surgical assessment in some patients in whom we need to localize language function and epileptogenic focus before the surgery. However, it works pretty well in the pediatric population. There are still some children who don't have access to the pre-surgical assessments and therefore to surgery because they are not able to perform the language task during the recording. So that's a problem for them. So we were thinking, we thought using resting state functional connectivity NIRS-EEG to see if we can localize language function as well as with the task. So we recruited 33 LT participants from 3 years old till adulthood, and they all did a FNIRS-EEG recording at rest and during a language task because we wanted to compare both conditions. We used a seed-based approach in this study. So we can see here each letter, A, B, C, and D, is one participant. So we have a 3-year-old girl, a 10-year-old boy, 11 years old, and an adult. We can see here in the first column result from mapping from functional connectivity NIRS, and here is the task-based result, and the same thing here. And we can see that there is a very good concordance between maps, between the functional connectivity and the task-based. So when we compared the overlapping of maps between the two conditions, we found 85% of participants had a good concordance between maps, and the language laterality index also was good between both conditions. So now we want to push this a little further and to see if it's applicable with children with epilepsy. And also we want to use different approaches. So now we use ICA approach and the ParaFAC approach Pedro introduced before. So these are really preliminary data because we just have 8 children with epilepsy so far. This is the work of a PhD student in my lab, Alejandra User. And so as I said, there are only 8 children, but so far this is promising. So we are encouraged by the preliminary result that show that there is the same kind of concordance and in a healthy participants. So maybe we will be able to do rest and state functional connectivity NIRS-EEG for the pre-surgical workup of very young children, younger children, and children with severe disabilities. So what's next with the FNIRS-EEG? First, we are now applying this in other pediatric population. Many teams work on FNIRS-EEG in preterm, for example. We are now applying this also in babies with congenital heart disease or in people who suffer from malnutrition. And But we also need to develop more accessible and user-friendly NIRS-EEG device, helmet, and software. So as I told you, many labs are developing their own helmet, their own device, but it's very time-consuming, it's energy-consuming, it's money-consuming. So hopefully there will be more of this on the market, available on the market. And finally, I think there are many developments that will come soon, and we need a lot of data analysis tools to better integrate both modalities. So using the rich information from both signals, the neuronal and hemodynamic signal provided by both techniques. For example, we could use the EEG to better predict and model the hemodynamic response. So just to conclude, there will be a special issue coming by the end of the year in Frontiers in Neuroscience about the combined EEG and NIRS, so looking forward to reading it.